Today I want to talk to you about some research project that has been mainly investigated by this guy, Stefan Peichel, who's a former postdoc in my lab and now a group leader in our institute. And so with him, we have been investigating a bit the effect of range expansions on functional genetic diversity. And because in our lab, we have been interested for a long time about the effect of range expansions on neutral genetic diversity. But right now, we are focusing more on this diversity that is under selection, and that will be a bit the topic of my talk. So, as you know, range expansions are relatively important. Have been driven in many species by some kind of change in the climate over the last 20 or 50,000 years or so. And so, for instance, in Europe, you had, during the last glacial maximum, many taxa that were located in some refuge area. And when the climate got better, then these taxa re-expanded north, could be plants, but animals as well, and probably humans as well. So this is not the only kind of range expansion that can happen, but you can have these things happening over thousands of years. But in the case of invasive species, then these expansions also occur over much more recent periods. So I think there is a general interest to understand a bit what are the consequences of these expansions that have been potentially touching every species. Also, if you think about every speciation events, usually these speciation events occur in a given location, and if a species has expanded its range since its ancestral species, then it must have also occurred through some kind of range expansion. So in our view, almost all species on the planet have been expanding their range at some stage. So that's a bit why we think that this phenomenon is important. So like maybe 10 years ago, we have been a bit looking at the effect of these expansions on patterns of neutral diversity, and this followed a bit some kind of work by Edmonds and Sforza, who studied a bit the fate of new mutations occurring on expanding wavefront. And so what you have on this slide is just a representation of an expanding population that expands its range from the left to the right, and here we can a bit represent this population as a collection of genes that, and in that case, you have two types of genes, so the red and the green genes, and just by chance, on the very edge of the front, you have some kind of red gene, and if the expansion proceeds by just randomly sampling individuals that are on the leading edge to colonize new territories, then you have a higher chance to draw some individuals that have these red genes than the green genes. Okay, so if you go on with this process, it could be that this red gene would just increase its frequency, but just completely by chance. Okay, and in that case, this gene will go potentially up, reach very large frequencies, and even reach fixation. And so what you see is that this gene has been what we are calling surfing on this expanding wave of advance, and that's this gene surfing mechanism that we have been describing. So if now we look on top of this expansion, so suppose now we have an expansion in 2D that goes from left to the right. This arc of circle represents the expanding front, and suppose the mutation just occurred at the wavefront or very close to it, then just by chance, this new mutation can expand its territory and become very frequent in these newly settled territories and even reach fixation here. Okay? So this phenomenon of this fate of new mutation was first described by Sforza, and we've been following a bit on this to detail a bit this phenomenon a bit better, but somehow what you can see is that these mutations, some of them can travel with the wave of advance. It's not something that occurs deterministically, but maybe a few % of all these new mutations will surf in this way. This phenomenon is not restricted to new mutations. It can also occur on standing variations. So any kind of allele that is on the wavefront can also go to fixation just by chance. So what also is interesting to see is that this mutation that used to be rare can become extremely frequent, and which somehow can mimic a bit the effect of selection. If you think of selective sweep, normally if you have some selected variants, that's a bit what you would expect under positive selection to have this kind of spread. So somehow in this range expansion, you can have phenomena that looks very much like selective sweep, even though they are completely neutral. Also, something interesting for people interested in phylogeography, what you see is that the place at which this mutation is currently the most frequent is not the place where this mutation originated. Okay, so if you want to base information about the origin of mutations, if your population has been expanding, you should take into account this expansion wave to try to interpret correctly the place of origin of these mutations. Okay, so that's something that should happen for neutral mutations, but actually not only neutral mutations can serve, but advantageous mutations can serve as well, and of course, and also deleterious mutations can occur. And on this slide, I just wanted to show you the fact that the spatial distribution of these mutations does not really tell you what kind of process are generating these mutations. So because you can have exactly or very similar geographic distribution of these mutations at a given point, even though they have been obtained for different types of mutation. Just that the place of origin of this mutation would be different in the three cases, but usually you just observe some distributions, but just by looking at distributions, it's difficult to know if it's an advantageous, neutral, or deleterious mutations. Okay? But the interesting thing was also to see that these even deleterious mutations can serve, which is something that maybe was not really extremely well known. So to study a bit better what happens during an expansion when you have a mixture of neutral, beneficial, and deleterious mutations, we first started by using some simulation approach. And a few years ago, we published a paper where we described a very simple model of expansion, where we assumed that we had some stepping stone model, and we had a system where we had a few populations that were connected by migrations that reached some kind of mutation, selection, migration equilibrium. Okay? And at some point, we allowed this population to expand its territory and to colonize some new areas. So they were sending some migrants to colonize these new areas. And then we assumed that we had some kind of local logistic growth of this population, and then some migrants were further sent away to colonize new environment. It's just in a stepping stone manner. So we could continue this kind of process. So this is a bit the assumptions of our model, so that there was local logistic growth. We also assumed that these mutations were drawn from some kind of distribution of fitness effects. In sample cases, we just assumed a very simple bimodal distribution of fitness effect. Just some fraction of mutation would have would be deleterious with a selection coefficient minus s, and some other fraction would be advantageous with selection coefficient plus s. Okay? And we would allow for actually more slightly deleterious mutations than beneficial mutations, as you would expect in normal coding region sequences. Okay? So we also assume that some kind of multiplicative fitness overall loci, we assume no dominance, no epistasis, and so far in this kind of model we assume some soft selection so that the carrying capacity and the growth rates were not depending on the fitness of the individuals or the populations. Okay? That's a bit the basic assumptions in our model. So what we obtained very typically was a bit the following. So on this graph, you have on this axis the geography, space. So we start from the left and we colonize, we go to the right. And on this axis, it's time. And the color code is fitness. Okay? So we assume that we start with a fitness of 1. So we scale the initial fitness to 1. And we see that if we follow this line, it means that we are following what happens about the fitness of the population in the core. And we see that this fitness is increasing because we have some positive selection going on and series of selective sweeps, and then the beneficial variance will fix in the population, increasing the fitness. Okay, so that's what we have. But now if you follow this line, it's how the fitness evolves on the front. Okay, and here it's very different. So what you see is that it starts with a fitness of 1, and then it gets more and more blue, which means that the fitness is decreasing. Okay, so And so what it means is that during the expansion, the fitness on the front is decreasing, and is decreasing with an extent that is proportional to the distance to the front. So if you look at any time point, you will see that you have a gradient of fitness from the core, the origin of the range, to the edge of this range. Okay? And that this fitness, this gradient is basically increasing over time. Okay? So you will have in the core positive selection going on, and on the front, basically this, what we will see that we have some kind of accumulation of deleterious mutations. Okay? In this case, we have 10% of beneficial mutations and 90% of deleterious mutations. But they have the same effect, opposite effect, but we just assume this. Okay? So, and so what it implies is that these new mutations that occur will be accumulating over on the front, but they accumulate almost at the same rhythm as they appear, because there is very little selection on the front. And so we have called this phenomenon the expansion load, because it's some kind of mutation load that is created by the expansion. So what happens, basically, is that on the front, since you have very low densities, it means that genetic drift would be very strong. Okay? So it means that selection would be very inefficient, and mutation will just accumulate at the same level as they appear, unless they are extremely deleterious or lethal. Okay? So you have almost no selection on the front during this expansion process. Okay? So this phenomenon does not only occur in one dimension, but also in two dimensions. So it's a simulation of a radial expansion from the center of this square world, and then you can see but in this case you have a bit more heterogeneity. But if you will follow any kind of line from the center to the edge, you will see that globally this fitness will decrease over time. Again, we start with a fitness of 1, and in the core it becomes more red, so it increases, but in the in the edge it's bluish, so it has decreased. Okay, so it's not something that is restricted to one-dimensional, but it's also two-dimensional expansions. And on this slide here, you have somehow the frozen record of the fitness during the expansion on the front. Okay, so it just means in that case that you started with a fitness of 1, and you can follow any kind of axis of expansion, and you should have a global decrease in the fitness, also in this two-dimensional space. Okay, so globally, this expansion load also occurs during these 2D expansions. In that case, here we have a two-dimensional expansion, but just more along one axis only, not a radial expansion. And here the fitness, how it evolves on the front. Each of these dots is the fitness of one dim that occurs on the front here. Okay, so it globally decreases over time and over space. So we think it's relatively general process, not limited to 1D expansion, also occurs in 2D. The difference between 1D and 2D expansion is that fitness is recurring much faster in 2 dimensions than in 1 dimension. Yeah. Right. Okay, in this case, I think the currency was 100 individuals per dim, which is not unreasonable. You're right that the larger the local population size, the more selection should be active, but these kind of things still holds until you have currency in the thousands. We didn't try with tens of thousands because these are individual-based simulations, and so we can't really go up to more than 1,000 or a few thousand individuals. Okay. Right. Right. I think that no, it does not depend on the mutation rate. It depends on the ratio of the beneficial over the deleterious mutations. So what will determine if we have this accumulation of deleterious mutations is, as we shall see in a few moments, that this fraction of deleterious mutation must be a bit more than 50%. Oh, right. Right. But it's just you need just to rescale the time, and then you will have the same phenomenon. It just will take more time to occur, but basically it's just a rescaling of the evolutionary time, you know. Oh? Okay, so of course for fast evolving organisms, this mutation, the extinction load will be more pronounced for very slowly evolving organism, but it should still occur. Then, after having a bit studied this model of self-selection, we turned to the question of what happens if we have hard selection. So in that case, by hard selection, we mean that at least in this model of hard selection, that the carrying capacity of the population depends on the average fitness of the population at any given time. So we start with a given initial carrying capacity, and then, depending on fitness, the carrying capacity will potentially decrease if the fitness decreases. And same thing for the growth rate of the population. Okay? So that's a bit our model of hard selection. There are other models where you could have only carrying capacity that would be touched or only growth rate that could be touched. But basically, in this case, we varied the two types of effects. And actually, we also studied what happens when you have recombination or no recombination. Okay? So this is a typical simulation that you get when you have no recombination, single locus. And same thing, this is geography, this is time, and here the color code is a bit different. So the more reddish, it's higher fitness. The more blue, it's again a lower fitness. So what you can see in that case is that you, again, tend to accumulate deleterious mutations, but which implies that you have a decrease in the fitness of the population, which affects actually the growth rate and therefore the speed of the expansion. And the speed in this absence of recognition can even stop completely until the front is recolonized by some higher fitness mutants that would invade the front, initiate a new bound of growth. But during this growth, they will again accumulate mutations, and at some stage, the expansion will stop again. Okay? So on this graph, you have a bit some replication of this evolution of the fitness, and this solid black line represents the average fitness over 100 simulations, and these different curves in different colors represent just individual simulations. And you can see that you have some kind of variance in the evolution, but you overall follow this kind of pattern relatively nicely, this nice decrease, until you reach some kind of equilibrium fitness, where that is actually due to the fact that the expansion has just stopped. Okay? So what you have in this case, in absence of recombination, is you have this kind of punctuated expansion with a long period of stasis and suddenly some new bounds of expansion that is initiated by the invasion of the front by a fetal lineage. Okay? So what is interesting on this slide is to see that in this case, which is where we have a completely homogeneous environment, the range of this species can be just limited by the mutation load. Okay? And it is until now, this potential explanation had not been considered in explaining why the range of species could be bounded. Okay, that we don't mean that everything happens like this, but that's also an explanation for why, let's say for non-recombinant organisms, the range of these organisms could be limited in space. Now with recombination, we have something different, a bit similar, but not quite. What we see is that we also have a decrease in the fitness, but in that case, the fitness decreases, reaches some kind of equilibrium value again, but the expansion proceeds at a slower speed. Okay, it doesn't halt at all, but it just proceeds at a slower speed that seems to be constant over time. Okay, and I'll come back to that when I will present you some results on some analytical model where we think we understand what's going on. But the situation is a bit different. It doesn't hold completely. So with recombination, you can also see that the fitness increases much faster in the core than in the case of no recombination. But the expansion wave does not stop completely, but continues at a lower rate that seems to be constant over time. So what we don't know when we just look at these simulations is whether this equilibrium speed is maintained by some kind of migration selection equilibrium. You could imagine that some larger fitness individuals invade the front and restore the fitness a bit, or is it something that is just due to something that is purely occurring on the front and without intervention of any kind of migration from the core? And we'll have a look at that in a few slides. Okay, but even with the important point here also is to see that with hard selection, we have a bit the same thing as with soft selection. We have globally decrease in the fitness, but in that case, we are reaching some kind of equilibrium value that we don't reach with soft selection. So again, this is more like evolution with hard selection in 2 dimensions. In that case, no recombination. In that case, with recombination. So, and this represents a bit the current range of the species and its fitness at different time intervals. So we have a regular increase. So you can see that between this point and this point, the population has progressed by this amount. For the same amount of time, later on, it has progressed this amount. So you can see that also in that case, the expansion slows down. Okay? And with recombination, it also slows down, but it globally goes faster than with in absence of recombination. Okay, so this 2D expansion also slow down in models of hard selection. So now to understand a bit better what's going on, we looked at a bit at the composition of the load on the front. So on this graph, that's the evolution over time of the fraction of the mutations that are deleterious that originated on the front. Okay, so of course at the beginning of the expansion there was no front, so there's mutation that occurred on the front, and as long as the expansion progresses, then this fraction of these mutations will increase, but will reach some kind of plateau and begin to decrease. It will decrease slightly with recombination and will decrease sharply without recombination. Okay, so what it suggests is that in that case, if it decreases, it implies that some mutations that did not originate on the front are actually found on the front after some time. So it implies that some mutations that used to be in the core migrated to the front. Okay? And they migrate much easier, in a much easier way if you have no recombination than if you have recombination. And we have actually exactly the same kind of process if we look at beneficial mutations as well. So it seems here that in our model, of course, we have still interactions between the core and the front, okay? Which would suggest that this equilibrium speed for the case of recombination could be maintained by this migration selection balance. Okay? So to study that a bit in more details, we developed some kind of analytical model that is a bit of a simplification of the simulations. So in this model, we assume that we had time there and space. Space is in here, actually, and time is here. So we assume that we start at any given point a new DIM at a given number of with a given number of founders, and then we have an exponential growth in that case until we reach a current carrying capacity. And once we have reached this carrying capacity, we colonize a new dim, and we send some migrants to this new dim. Okay? And so we have this kind of process. We grow, reach carrying capacity, and this stage we colonize the new dim, and we again begin this kind of growth process. Okay? It's a very simplified view of this simulation model. And in that case, we are just following what happens on the front, and we assume that there is no migration anymore between the core and the front. Okay, so it's a bit different from what we had with our simulations. What is important to consider is that in our case, we have just selection occurring during this kind of growth phase. And again, we have this model of hard selection where we have the carrying capacity and the growth rate that depends on the local fitness. So typically what happens is that the carrying capacity will decrease over time, okay? Because the fitness will decrease over time. And also what is important is that the time it takes to grow to carrying capacity will also increase over time because the growth rate is becoming lower because the fitness is becoming lower. Okay? So that's a bit the characteristic of this analytical model. So relatively simple. So I will spare you the details, but under this model, it's possible to get the property of fixation of new mutations. And this is this property of fixation. It's relatively simple, and what is interesting to see is that it has exactly the same form as the property of fixation that you would have for in a statutory population. Okay? But in that case, this effective size is just replaced by this factor f times t, and f is the number of founders that that founds the new dim, and this is equal to the carrying capacity by times the migration rate divided by 2, and times the t, the time it takes to reach this carrying capacity. Okay? So otherwise, that's exactly the same kind of form. But what is interesting is that if you contrast this fixation probability in the stationary population, which is this gray line, we see that this fixation probability increases very quickly and reaches this 2s probability when it's positive, and it decreases very fast for negatively selective mutations. But we can see that on this expansion front, that's a bit how it varies over time. So the black line is the analytical solution, and these bars represent replicates obtained by simulation under our more complex model that includes migration between neighboring dims. Okay, so we can see that we can first see that our approximation is relatively good compared to a more realistic model under simulation. Okay, but what's important to see is that beneficial mutations have fixation probability that is maybe only 2 times more than neutral mutations, and maybe 4 times more than deciduous mutations. Okay, so they still have, and these deciduous mutations have a non-zero probability to go to fixation if they occur on the way expanding front. Okay, what is also interesting is that we can find conditions under which we will have this expansion load. And what we mean when we have expansion load, it means that we have a decrease in fitness between generations. Okay, and here we can condition that for different things, but for instance, this phi d represents this proportion of mutations that are deleterious over the total number of mutations. Okay, and we can see that we will have expansion load if this quantity exceeds this value, which for very small values of this 4 FTS value is just 1/2 plus this thing here. Okay, so in our previous simulations, we would have had expansion load if the fraction of deleterious mutation would be more than 54%. Okay, and of course, the higher the section coefficients is, the more deleterious mutations you need to have this expansion load. Okay, just also to show you how good is the fit between simulations and analytical results. So we have, for different migration rates between neighboring dims and different section coefficients, the dashed line is the analytical expectation, and the solid line is the average over simulations. So we see that if we have very small section coefficients, we have very good fit with between simulations and analytical predictions. If we increase this selection coefficient, like 2.5%, which is relatively high, then the fit becomes not so good, but still more or less okay. But if we have a very high migration rate, then the fit becomes very bad. Okay, but our analytical prediction is always conservative in the sense that the decrease in fitness will be always faster than what we would predict with the analytical prediction. Okay, but anyway, it's of course the more migration we have, the more difference we have between our simulation model and our analytical model. So now if we go back to this example here, so where we had actually this kind of constant but slower expansion speed that we obtain here, when actually it's at the point where actually the fitness remains constant over time. What seems to happen is that since we have relatively good fit between what we see, which is this solid line and our expectation under this model, it suggests that actually this reduced speed is only due to what happens on the wavefront, and it's not really due to the fact that we bring some beneficial mutations from the core to the front. Okay, so what we think is happening is that when the expansion proceeds, we accumulate these mutations, which slows down the expansion that we see. Then it makes selection more efficient because the time between consecutive period of conization increases, so selection has more time to operate, so it becomes more efficient. And it means that if selection is more efficient, it will better purge these deleterious mutations. Okay? And if it purges mutations, then the fitness of the population will increase again, which allows the population to go a bit faster, and then we have this kind of negative feedback loop that leads to some kind of equilibrium speed that we see here. Okay, so this equilibrium speed, we think that it's something that is only due to the properties of selection going on the front. Okay, and we can actually measure what is the equilibrium speed, which is this value here, and anyway, so we can show actually that this value here is actually larger than the speed of the spread of beneficial variance in the core. That would be just the square root of 2ms. And for this example here, this log r not w bar is just 2 times s. Okay, so anyway, we can find some conditions for this speed. Okay, so that's a bit the end of this part of my presentation, and now I would like just to turn to a new aspect. Is so far we just assumed that all the mutations were codominant, and now we just want to explore the case where actually we have recessive mutations. Okay, so that's a bit what we would expect for the evolution of the fitness with codominant mutations. That's a bit what we've seen. So here it's the decline is relatively small because we are just looking at a small evolutionary time period. But if we now look at what happens with recessive mutations, it's something like this. So we have a sharp decrease in the fitness in the initial phase of the expansion. And why do we have this? It's just that because for recessive mutations, we used to have very small frequencies. And during the expansion, you have the creation of, you have random genetic drift, and then some of these recessive mutations will disappear. And because the overall frequency of these recessive mutations remains the same due to this recurrent bottleneck, it implies that the frequency will increase at some other loci. And it therefore implies that some homozygotes will be created during this expansion process. And in that case, of course, because we have these homozygotes, the fitness will decrease quite dramatically. But after some time, we will reach again some kind of equilibrium value, and we'll have a bit this decrease that would be expected under our model. Okay, so with recessive mutations, we have initial very sharp decrease in the fitness, and but after some time we get some evolution that is close to what we would have expected for our previous model. So in 2D, we have something a bit like this. Before it was like one-dimensional expansions. Again, same kind of things happen. So you have a decrease in the fitness. In this case, the color scale is a bit reserved. If it's red, it means lower fitness. You see that in that case, with recessive mutations, you have a bit more heterogeneity on the front about the fitness. And you can have some places in the front where the fitness is relatively high and very low for the places on the front. But still, you have this decrease in fitness, but with more heterogeneity of fitness on the front than in the case of codominant mutations. And if we really focus on the fitness on the front, what happens? So over 10 simulations, here we have 10 curves that represent the fitness of population that goes in this direction. So for this particular simulation here, that would be how fitness would change. So very low fitness on the edges and large fitness in the middle, and then again some low fitness. But it's not always that the higher fitness is in the middle. You know, there are some cases where the highest fitness is very close to the edge. Okay? So there are just 10 replicates. What is interesting there is that if we look at what is the amount of heterozygotes that we see in this population, you have a very close match between the heterozygosity level and the fitness of the genes. Okay? So what happens is that you have a correlation between heterozygosity and fitness in case of recessive mutations, and this correlation is quite high actually. Okay? So we can look at this correlation for different replicates, and here I have these different color codes. So what happens on the front, you can see that here you have the mean fitness, here the heterozygosity. You can see that for different points of the same color, you have this very high correlation. But if you look at correlation between the fitness and heterozygosity in the core, then you have absolutely nothing. Okay? So this heterozygosity, this correlation between heterozygosity and fitness seems to be a property of the front of the wave. Okay? So now, since we have all these expectations about how fitness should evolve, we wanted to see, okay, do we see any kind of evidence of this expansion load in some organisms? So the organism that we've been looking at is humans, because we know that humans have expanded over time from Africa, and they have colonized the whole world. So this might not be the exact migration pathway that they have followed, but somehow they must have colonized the world in a kind of a range expansion process. Okay, so if we are correct, we should expect to see some kind of expansion load in humans. Okay, the problem there is that you have this is a very hot topic right now, and many people do not really agree about whether we see a larger mutation load in non-African population than in African populations. Okay, so there was an early publication by Kirk-Lohmüller who showed actually that non-African populations have proportionally more deleterious mutations than African populations. Okay, but since then you had a few papers that actually don't find differences in the load when measured as if mutations would be codominant. Okay, so the number of deleterious mutations per individual seems to be pretty similar in Africans or in non-Africans. Okay, so and all these type of recent publications seem to see this, even though some actually would see a slightly more mutations in non-Africans, but you need extremely large sample sizes to see this stuff. Okay, so to look at that, we collaborated, or we were included in a collaboration led by Carlos Bustamante and Brenna Hen, and where they actually resequenced the exome of 53 individuals from 7 different populations that are found, some in Africa and some a bit outside sub-Saharan Africa, so at different distances from Africa. Okay, so this looked like a nice way to test whether we find this kind of cline of this mutation that we would expect under our model. Okay, so to have a look at this pattern, we try to better characterize the effect of different mutations that we would see in these exomes. And for this, we used an index of, or a score of mutation conservation that is called this rejection substitution score. That is basically the difference between the observed and expected number of mutations that have occurred on a given phylogeny of mammals. Okay? These people just studied 35 mammal species, look at syntenic regions, and they assumed that they knew what was the divergence time between all these species. They would know the branch lengths, and if you know the mutation rate, you couldn't predict more or less how many mutations would be expected on a given branch. So if you see less than what you would expect, it implies that these mutations have been removed by selection. If you see more, then they have been accumulating faster. Okay, so and the fewer you see, the more conserved this site would be. Okay, that's a bit this kind of RS score or GURB score. So what we did was and that's a bit the distribution of these number of sites that have different GURB scores. So all GURB scores between -2 and +2 are supposed to be close to neutrality, and something larger than 2 would be conserved. And between 2 and 4, we assume it's moderately conserved. Between 4 and 6, it's largely deleterious. And if it's larger than 6, it would be extremely deleterious. Okay? And again, you have fewer and fewer mutations that would have very large effects on the fitness. Okay? So we don't know exactly how these GURB scores relate to selection coefficients, but we could imagine some kind of degree of proportionality. Okay? So what we did was just to plot a bit the number of mutations per individual that would have different GURB score, and that's a bit the kind of thing that you get. Now, if you look at heterozygosity for neutral sites, so the GURB score between -2 and 2, that's a bit how it varies for all the 53 individuals. These are the 2 African populations, and heterozygosity for these neutral sites declines with distance from Africa. Okay? And Africans have more heterozygosity, as you would expect, since these populations have gone through bottleneck and then this expansion. So now if you look at mutations that have different GURB score, we can see exactly the same pattern for moderately deleterious sites or very deleterious sites, but we don't see this pattern for extremely deleterious sites. Okay, so for these extremely deleterious sites, we have no decline at all, and it seems that this heterozygosity level is very similar in different populations. So now if we translate this into a measure of load, and actually if we just look at the number of homozygous sites per individual, of course, you see the would just be how homozygosity would change. It's the inverse of heterozygosity. It would increase. But when you have selection, this number of homozygous sites would be a measure of the recessive load. Okay? So we can see is that this number of loci that are homozygous will increase actually with distance from Africa for this category and for this category as well, but not for this category. Okay? And again, here it seems that for these extremely deleterious mutations, there is some kind of fixed limits to the number of sites at which you can be homozygous for the extremely deleterious mutations. Okay, it's around, in that case, 25 to 30, and if you exceed that, maybe you don't survive. Okay, so so it means that if we assume that some mutations are recessive, then we have this evidence that there is an increased load with distance from Africa, which fits our expectation. Okay, and what is interesting, it also fits a bit our simulations. So that's again simulation-based thing. So if you have some expansion for a few generations, that's a bit how heterozygosity should decrease over time. That's how the homozygosity for the new mutations should increase over time. And what is also interesting is that would be the derived allele frequencies, that's how it changes. So basically it doesn't change at all. So if you were to measure fitness as if mutations were codominant, it would imply that you wouldn't see any difference between populations close to the origin or populations that are far away from the origin. Okay? So basically, it somehow reconciles a bit the previous observation that people said that, okay, there is no difference in the total number of alleles that an individual, total number of deleterious alleles that an individual has in Africa or away from Africa, and that's actually what we find because we see that this frequency of derived alleles do not change over time. What is changing is just the packaging of these alleles into homozygotes or heterozygotes. Okay, so if we assume that these mutations are recessive, then it should imply that there is some kind of expansion load. Okay, and maybe the last slide here. This slide actually shows why we would not really expect to have any expansion load for extremely strongly diffuse mutations. So I showed you that we can have expansion load if we have given excess of these mutations compared to beneficial mutations. But we can also see what is the condition under which we have expansion load for different effects of these mutations. Okay? And we have actually a condition that is looking like this, that is plotted like that. Okay? So here on this axis we have the number of founders of, that are used to found the new DMs during the expansion, which is proportional to the migration rate. And on this axis, the effect of the mutation. So what we can see, if we are below this curve, we'll have expansion load. If we are above this curve, we will not have expansion load. What it means is that for a given migration rate and carrying capacity, if we increase the selection coefficients, then we will not have expansion load. So we'll not accumulate these mutations over time and over space. Okay, which is exactly what we've actually have found in this case. So for the extremely deleterious mutations, then we don't expect to have this increase over time and space of the load, but we think that if we decrease the effect of the mutation, then of course we will have this expansion. Okay, so it exponential growth, yes. The time it takes is the time during which selection acts. No, it's time to reach the carrying capacity from the number of founders. Okay, and this goes down over when the expansion proceeds because the fitness decreases. Okay, so that this result somehow explains why we would not have any kind of expansion load for these very deleterious mutations. Okay, so I think I've reached a bit the end. So I hope that I could show you that these deleterious mutations are expected to accumulate during the expansion, a bit like neutral mutations. So it's a pure neutral process. So they accumulate because on the front they behave as if they were neutral. Then this process that is driven by the front, actually it's not only about the front, but it's about the whole range of the species, because at any time point, a given part of the range used to be the front. Okay, so this gradient in fitness, I didn't show you that, but it persists over hundreds or thousands of generations after the range has been fully colonized. Okay, so it's still a transient effect. It will eventually disappear, but it takes a long time to disappear. So we should still see this effect quite long time after the end of an expansion. Then I showed you that this expansion was robust to recombination. That I didn't show you, but we had simulation where you see that. So even if we have long distance dispersal, we still see this kind of expansion load building. Also didn't show you that, but even if we have a bit more complex distribution of fitness effects rather than just this bimodal distribution, we still see this effect. And if we have hard or soft selection, we also see this effect. Of course, the exact dynamics of the building of this expansion load will depend on the exact way, on the exact parameters, whether we have recombination or no recombination, but somehow expansion load is expected to occur unless we have extremely strongly deleterious mutations. Okay, and concerning human populations, I think we have some evidence that we could have some expansion loads created if we assume that some of the mutations are recessive. If they would be codominant, we don't see any kind of signal of expansion load. And the only thing I want to be cautious about is that even though we see that there could be some kind of expansion load building, it's very difficult to translate this measure of load into a measure of the fitness of the population, because all our stuff is still based on, or still assume that the environment is constant, but of course, environment in Africa and the Americas is quite different, so we can't really conclude that Africans are much fitter than Amerindians or this kind of thing. So it's a bit more complex than this. Okay, so, and this is the people I would like to thank. So Stefan Peichel, who's been really the main driver of all this effort. Isabelle Dupré for assistance with genomic data, Mark Pratik for modeling part, and all these people for providing us access to this nice data set on human population, and our funding agency, and you for your attention.